Hey y'all, for my final project I decided to do a paper about kinetochore oscillations involved in chromosome segregation. So the paper I'm doing today is called Super Resolution Kinetochore Tracking, Reveals the Mechanisms of Human Sister Kinetochore Directional Switching by Burroughs et al. Whew. Anyway, that's all just a fancy way of saying bougie videos of the kinetochores show how they move around. So I like this paper a lot because of the cool figures and models they present, but before I go into specifics, I'm going to start with why I chose this paper. Let's talk about my personal connection to this topic. The main connection is that this is part of what I study in my independent research. In my lab, we're more so focused on proteins involved in microtubule attachment to the kinetochore, specifically right now the NDC80 complex. It looks like this. It has 4 proteins involved, and the one I focus the most on is HEC1. This protein has a tail region that worms its way into the microtubule to anchor it to the kinetochore. So we've done a lot of cool mutations on this protein, but one of the coolest things that my lab has me thinking about is how dynamic this system is as a whole. Something I feel is barely covered in our college education is just how much these biological processes move around. So microtubule binding during cell division always seemed like a quick process of the microtubule grabs a chromosome, the sister chromatids split, the cell pinches and divides. That's it. In reality, the microtubules have to search for the chromosomes, the sister chromatids have to oscillate to generate enough energy to split, and the cell has to coordinate so many motor proteins to fully divide. So, although I have more of a background in microtubule binding of kinetochores, this paper focuses on this sister chromatid oscillation I mentioned before. By this I mean the microtubules pull the sister chromatids back and forth by the kinetochores over and over and over again until eventually the tension between the sisters is enough to make them go their separate ways. One of the biggest mysteries here is how the sisters know which direction to go in, when to change direction, and ultimately how this all is coordinated across every chromosome within the cell. It's a lot. This paper aims to get us started in this knowledge by focusing on which sister changes direction first, and by attempting to determine the mechanism that causes the switch in oscillation direction. They do most of their science through imaging live kinetochores, and then by using MATLAB to go frame by frame through the videos of the thousands of kinetochores they looked at to determine trends in which sister switches first. It's all a lot, but it makes for good science. So anyway, let's get into the meat of the paper. So the paper starts with the currently accepted model of how the sisters sort of decide when to change direction. The model suggests that the sisters change direction when the space between them has low tension. This occurs when the leading sister changes direction first, shortening the space between them. Then this promotes the trailing sister to change direction. This model is heavily reliant on the tension itself being the factor that makes the sisters change directions. And the paper ends up proposing a clock mechanism instead that deals with the switch timing. But we'll get into that later. Moving on to figure 2, they use this to characterize how they did their imaging, as well as how they constructed their standard models. So for imaging, they went through each plane of the cell in a stack in order to form a 3D image. In these pictures, they could detect kinetochores and then track them. This tracking data could then be put through whatever mathematical algorithm they used. From this data, they were able to generate a 3D image showing kinetochore locations in relation to the metaphase plate, the center of the cell where division occurs. They also were able to measure the trajectories of each sister, as well as the angles the sisters were displaced at from the metaphase plate depending on their distance from the plate. They also took these really cool videos that show how they were able to see the kinetochores in 3D space, then model them as points, then follow the path of those points, and ultimately they could track them against the metaphase plate. So this all seems like background for the rest of their work. On to figures 3 and 4, they take a look at what the distance between the sisters is before and after switching directions. This is dependent on whether or not the leading sister or the trailing sister changes directions first. This is a common theme in the paper, leading versus trailing, or as they refer to it, lead-initiated directional switching, LIDs, or trail-initiated directional switching, TIDs. This is all characterized in figure 3. Then in figure 4, they show that when the leading sister changes first, there is a lower distance between the two sisters, like this, and when the trailing sister changes first, there is a higher distance between the two sisters, like this. Since the distance between sisters has a positive correlation with the tension between them, LIDs have lower tension between them compared to TIDs. Also, in figure 4, they begin looking for trends in a cycle of lids then tids, or of tids then lids as the chromosomes oscillate. Whether or not you come from a lids into a tids or vice versa seem to play a role on the overall tension between the sisters. They found that tids overall have a lower inner sister distance regardless if the previous switch was a lids or a tids. This can be seen at the end of the graph in 4b. 4c and 4d are standardized times that sort of zoom in on the first half of 4b. They show the consistent spike in distance between sisters in tids right after switching direction versus a consistently shortened distance between sisters in lids right after switching. They stated that besides the changes in tension between the sisters, the dynamics are practically the same. It also seems that the movements seem to be very repetitive, in that many of the kinetochores reached the same peaks and valleys, and that most of them had the same runtime between switches. All of this consistency and repetition led the scientists to come up with some sort of clock mechanism being the underlying cause of the switches. Before going into their clock model, we'll take a look at one last figure though. So, in figure 5, they focus on the way the sisters twist away from the metaphase plate as they oscillate. They notice that as the distance between sisters increases, there is an inverse relationship with the amount of twist. So, tids with higher tension have smaller angles of twist, versus lids with lower tension have larger angles of twist. They liken this relationship to the action of a spring, where the high stretch can align the sisters better while they are under more force, versus a low stretch allows the sisters to move more under thermal and mechanical fluctuation. Again, we see similar dynamics being maintained by the tids after lids or tids, and by the lids after lids or tids. The way these dynamics are conserved also plays into their mechanical clock hypothesis. So, figure 6 finally lays out their proposition for a new model of kinetochore oscillation switching. They basically say that, rather than the tension between sisters being the key factor that causes the switch, there is a clock in place that tells the sisters when to switch. This clock is reliant on saving the process from catastrophe. So, if things are out of line, the clock will trigger a switch in order to achieve balance again. If forces level out, the clock will allow the oscillation to continue. So, in figure 6a, we see their diagram showing that both sisters have a clock in place to deal with the coordination of switching. The dotted blue arrow indicates that if the leading sister accelerates too quickly, it will switch first since the tension between the sisters is too high for a tids to cause even more tension. So if you think about it, too much tension too early on could result in an early splitting of the sister chromatids before cell division is ready, which could lead to chromosome missegregation. With that catastrophe taken care of, the solid blue arrow in the diagram shows that if the movement is stabilized, or if the leading sister is too slow, the trailing sister will move first to increase the tension in the system. This will help keep the chromosomes aligned with the metaphase plate. 6b shows that if you initially have a lids, which lowers the tension, but the leading sister in the next oscillation accelerates fast enough, the chromosome can handle another lids. Whereas if you initially have a lids and the leading sister doesn't accelerate enough and keep a balanced amount of tension between the sisters, a tids will occur to raise the tension and help maintain the balance between them. Initial tids show a similar trend that, if the high tension is lowered over the course of the oscillation, the sisters can handle raising the tension again with another tids on the next oscillation. This is where they show how the standard model from figure 1 is kind of wrong. So if an initial tids still has high tension by the end of the oscillation, a lids will occur to lower the tension between the sisters. This contradicts the standard model in that, over time in an oscillation after a tids, the tension is slowly decreasing. Yet, a lids can still be triggered from it. This shows that it is not the growing tension between the sisters that triggers the lids, but rather a high tension between the sisters that has been maintained for too long, allowing the clock to click into place and help balance out the system. In terms of the importance of these findings, I think that this is a great start in more kinetochore specifics. Learning how cells are able to manage chromosome segregation and regulate the timing of everything can provide a lot of insight on cell division. Incorrectly conducted cell division can lead to so many issues like cancers if conducted too often, or things like birth defects or even death if the DNA is not conserved correctly during fetal formation. So, although this is one small part of the equation, continuing to learn more about how cells can detect these tension-related boundaries and can make these decisions to maintain themselves can further our understanding in chromosome segregation-related health problems. Aside from just being interesting science, research like this is the groundwork for so many potential future projects. So, one thing I really liked about this paper is how well done their models are. Their figure 6 is easily legible and lays out their exact hypothesis clearly through flowcharts. One improvement to this model would have been some movement. So the model I'm proposing is animated. Just a simple model to show when ATIDs or LIDs would be necessary for the sister chromatids. This would pair nicely with their drawn model in figure 6, particularly figure 6b, and seeing as they've already included other videos for their 3D models, they could show a simplified 2D motion of the kinetochores they're describing. And one other quick way to understand why the chromosomes are oscillating the way they do is to use a household object to simplify the process. So take your two index fingers, each as a sister chromatid, and wrap a rubber band around them to signify the tension between the sisters. Now oscillate your fingers to the right and have your right finger move left first. This is a LIDS. You'll notice the rubber band lose tension and begin to fall, so to compensate, you'll then have to move your left finger in order to uphold the tension. From here you can experiment a little bit with the ways acceleration during oscillation plays a role in which sister switches first. So let's say you're moving to the right again and your right finger is moving faster than your left finger. You'll find that a lot of tension has built up by the time you reach the end of the oscillation. So if you attempt ATIDs by moving your left finger to the left first, you'll notice the tension only increases, making it a lot harder to hold on to the rubber band. Versus, if you were to attempt a LIDS by moving your right finger to the left first, you'll notice the tension decreases, making upholding the rubber band a lot more manageable. Feel free to play around with the rubber band and further your understanding of sister kinetochore directional switching. It's a really simplified way to visualize this pretty complex topic. Other than that, this concludes my presentation, so thanks for watching, y'all.